In the name of God, my name is Dr. Tayyeba Rastegar. I have PhD degree in anatomical science, and in this semester I tell for you some things of subjects of the histology. In this session, I want to speak about the introduction of histology and the general things that we should know that until we able to study tissue. In this session, you should know about the preparation of the tissue for study with microscope, include fixation and tissue processing, embedding and sectioning and staining. After that, I explain about different type of microscope and finally different methods for other studying of tissue. What is histology, or what is the definition of the histology? Histology include two words, histo and logy. Histo means tissue and logy means knowing. This science is a branch of the biology that we study about the tissue, the structure of the tissue with a microscope. I think, do you know cell? What is the cell? Cells are basic structures of the cells. This is the smallest living part of the body that have structures and function. When some cells and ECM or extracellular matrix of them comprise together the cells that have same structure and same function. This is very important. Yes, these cells and the extracellular matrix together comprise the tissue, and the tissue make up the organs, and finally organs form different system. At first, we should know that how we can prepare tissue or cell to be able to study them under microscope. For this procedure, at first we should obtain the specimen, gross examination of the specimen, fixed of the tissue, then we'll do the tissue processing, include the dehydration, clearing and impregnation, and finally embedding, staining and studying under microscope. Tissue fixation. What is the fixation? Fixation is a complex series of chemical events that differ for different group of substance found in tissue. In the fixation, we use some chemical or maybe some physical materials for fix of the tissue that prevent lysis of the tissue. Fixation is a reaction between the fixative and proteins that are in the cell membrane. So keeping everything as there in vivo relation to other and in vivo structure. The aim of fixation contain to prevent autolysis and bacterial attack. You know, in the cell, in cytoplasm of the cell, there are lysosomes that they are include enzymes that when we remove the tissue or cell from the body, this enzyme activate and autolysis or lysis itself occurred in the cell. To fix the tissue so they will not change their volume and structure and shape. Prepare tissue and leave them in the condition that we able to staining them in the session, and finally to leave tissue as close as their living structures as possible. And after that, able to study them. And also in this fixation, not a small molecule should be lost. For the fixation, we use some chemical materials such as formalin, glutaraldehyde, all different aldehyde that generally used in the pathology and histology laboratory. But some of fixation we do in our life, such as cryopreservation of some food in the refrigerator, or drying of some vegetables, that all of them are physical fixation that we do in our life for prevent of the autolysis and disturbing of different material. When we remove the tissue from the, for example, human body in biopsy in surgical operation room, or in the laboratory from animal laboratory, at first we fix them in the fixative, such as formalin or paraformaldehyde, that generally different lab use from formalin. These small basket are the location for putting of the tissue that, as you see here, have different type that we can close the door of them. We label the tissue in this step. For example, this is a liver, this is a kidney or other. Write by pen and put inside of this basket and close the door. And after that we can do tissue processing manually, but in most labs they do buy this equipment that named tissue processor that work automatically. It includes some jars, that each of these jars have these materials. These we can arrange it manually, and this equipment equally turn around this axis and finally finish between 18 or 20 hours. At first, the first jars include the formalin. Formalin is a fixative. This step is a fixation that we fix tissue. Include formalin. We put the basket that include the different tissues inside of this jar. After that, we should remove the water of the cells and tissue. Why? Because for the preparing of very thin tissue, very thin section, we need to cut the specimen as 5 to 10 micrometer. For this goal, we need to infiltrate tissue and cells with paraffin. You know, paraffin biochemically include lipids, and in the cell include the water. You know, water and the lipids are not affinity together. For this goal, at first we removed the water from the cell by ethanol that started from lower degree as 13 degree. After we increase the degree of them, 13, 50, 70, 96, and finally absolute ethanol. After that, the other step named clearing, that we used xylene that include 3 jars, that the tissue change from this jar, and finally we infiltrate the tissue and finally the cell with paraffin. In this step, the paraffin infiltrate to each part of the cell that previously include water. Each, and in the above part you see each time for each jar 50 minutes or maybe 1 hour 30 minutes each are changeable with this equipment, with this tissue processor. Here in this slide again you see different step of tissue processing. Step 1 is dehydration with alcohol that replaced water in the cell. After that, clearing is that we use the xylene. Xylene dissolves the alcohol, and finally in the last step, paraffin display xylene. And now the specimen ready for embedding or embedded in the paraffin. This is our specimen, and we put it in some basket again for embedding. This is a equipment for embedding, that this part contain the paraffin. The melting temperature of the paraffin is 58 until 60 degree, that this part include of it. We put the specimen here in some again basket as you see here, and then we release the handle of the paraffin. This part we put at first the sample. Here we should attend to the direction of the tissue. We want in the future, we want to prepare longitudinally or transverse section. In this part, in this step, we should identify the direction of the tissue. After that, we can reserve the embedded tissue for a long time, for example, for many weeks or many months or many years in the laboratory. After embedding of the tissue with paraffin, after that we should do sectioning. Sectioning with microtome. Microtome is equipment for cutting of the tissue as micron diameter. You can see here, we can see here a microtome, and this is a cryomicrotome. When we use the cryomicrotome, we use it for fresh tissue. We don't do the tissue processing. For example, in the operation of the human, the surgeon give to the lab a tissue, and for example, a tumoral tissue. He or she want to know that whether is tumoral or not. The technician of the lab section there by cryomicrotome, because in this procedure the tissue don't pass from tissue processor. They section them, and after that stain them. But normally in the histology and pathology lab, mostly use this microtome. This is a microtome that have one place for putting of the specimen that located in the embedded in the paraffin. This is a handle that we turn it, and in each turning the tissue pass from this knife one time and cut it in micron. And here we can regulate the diameter of the tissue, whether we want the 5 micron or 10 micron or 3 micron. Yes, we regulate by this, and after that we remove these section and put them on the slide. put them on the slide. You know the slide is very thin glasses. We put the tissue and attach the tissue on the slide by some glue such as albumin and glycerin glue, and after that in the laboratory the staining done. Here there are some jars that include some materials and also two of stain that generally used in the histology and pathologic laboratory, that is a hematoxylin and eosin staining. And this is a automatic stainer that in some lab maybe they do from this stainer. But in most of the laboratory, the staining done manually, that one person change them in different jar, and finally the tissue stain, and in the last steps the water that at first step we remove the from the cell again return to the cell by alcohol that decrease their degree. Hematoxylin and eosin is a general staining that used in histology. Hematoxylin have a blue, purple or blackish color. It stains the basophilic structures of the cell, such as different nucleic acids such as deoxyribonucleic or ribonucleic and ribosomes. And finally, the euchromatin DNA spread, diffuse and less stain, but the heterochromatin is a condensed DNA that stain dark, that I explained about this type of chromatin in the cell session. That, as you see here, hematoxylin stain the nucleic acid that you now are in the nucleus. Under microscope, the nucleus that stain with hematoxylin have blue or purple color. Eosin have a pink color. It stains the eosinophilic structures such as different macromolecules that are in the cytoplasm, such as proteins, such as collagen, and finally in the staining, the cytoplasm have acidophil or eosinophilic stain. In this table, there are different staining for different structures of the cell. The hematoxylin and eosin do generally in different for different tissue is a general staining when paired with eosin, or eosin is a general staining where paired with hematoxylin. The hematoxylin stain the nucleus as blue color, and the eosin stain the cytoplasm as pink color. And this is a staining also for other structures. For example, the eosin also used for the can stain the different fibers in the cytoplasm. But other of the staining are special staining that in the different lab use for some structures especially. For example, toluidine blue is another general staining that all of the structures of the cells is blue, and mostly used for fast staining or for mast cell granules. The Masson trichromo and Mallory trichromo staining done for connective tissue, for staining of the collagen fibers, and also for other structures as you see here. Weigert is for elastic fibers or reticular fibers, that is a type of the collagen fiber stained by silver stain. Wright for blood cells, and Orsein for staining that do in different laboratory. All of them are the special staining that do in some special procedure.